Welcome everyone to the IIVS-sponsored webinar on the KeratinoSens assay for identification of skin sensitizers. I'm Amanda Ulrey, your moderator and web organizer. We'll begin the webinar with an introduction to skin sensitization by Dr. David Basketer of DAB MEB Consultancy. This will be followed by an in-depth discussion of the KeratinoSens assay by Dr. Kimberly Norman, IIVS Study Director, responsible for skin sensitization work. We will close the webinar with approximately 15 minutes of question and answer with Drs. Basketer and Norman, who will be joined by Dr. Andreas Natch of Givaudan, developer of the KeratinoSens assay. Before we start, let's take a moment to ensure that everyone is ready and familiar with the webinar control panel. Due to the large number of attendees, all audio other than the panelists has been muted and will remain so for the duration of the webinar. You may ask questions by using the question feature at the bottom of the control panel to the right. Click on the arrow to maximize the control panel and view the questions window. Technical questions can be submitted anytime, but will be held until the end of the presentations when they will be posed to our panelists during the 15 minutes allotted for the question and answer period. Any questions that are not able to be answered within the scheduled webinar time will receive responses from our panelists and will be distributed to all webinar participants within a few business days. Lastly, we are recording this webinar and the question and answer session at the end. Let's get started. Today's first presenter is Dr. David Basketer. David is a Eurotox registered toxicologist who co-developed the local lymph node assay, the first alternative method for identification of skin sensitizers to undergo formal validation during his time at Unilever Safety Laboratories. He now undertakes freelance consultancy work in toxicology, including the development of non-animal methods for the identification and assessment of irritants and allergens. David currently chairs the European Center for the Validation of Alternative Methods Scientific Advisory Committee, as well as their Skin Sensitization Validation Management Group. Take it away, David.
Thanks, Amanda. That's the Basketer advert over, folks. Hello to you all, wherever you are. It's my job to give you a little bit of an introduction to this territory. And if I can move straight away to the second slide, you'll see that that covers the mechanism of allergic contact dermatitis. And for anybody who's not totally familiar with this already, forgive me, chemicals come into contact with skin. You see that on the top left. They have to react with skin proteins, cause a little bit of disturbance that triggers dendritic cells to migrate out of the skin down to the draining lymph node, where the mature dendritic cells then interact with various kinds of cells, including T cells. And if the T cell recognizes the chemical that's been brought attached to skin protein, they're stimulated to divide, recirculate around the body, and then you have the sensitized state such that subsequent contact can produce the clinical manifestation allergic contact dermatitis. Now, all our previous tests prior to the local lymph node assay were based on the guinea pig to identify chemicals that cause this kind of problem, and they all recapitulated this whole process, and we looked at redness on the flanks of guinea pigs to see if we could identify a sensitizer. Next slide, please. What this next slide shows are the protocols for the two commonest guinea pig tests, the Bueller test and the guinea pig maximization test. They take several weeks to perform, and I don't need to go into lots of details here. What we did then with the local lymph node assay was to look at a different way, next slide please, to look at a different way of assessing skin sensitizers by looking at the proliferation in lymph nodes that drain the site of application. And here you see a cartoon of the lymph node assay. It was a refined and reduced assay that found a lot of favor ultimately through validation and in regulatory circles, but of course it's not an in vitro alternative. Next slide, please. What you can see on this slide is a little cartoon of the world of chemistry, and I just wanted to introduce this concept to everybody because I think it's very relevant when we're thinking about validation of alternatives. The world of chemistry is the big oval, and you can see groups 1, 2, 3, and 4, where number 1 are the non-sensitizing chemicals, and 2, 3, and 4 cause sensitization. In fact, in regulatory toxicology, only groups 3 and 4 are the ones that end up being classified and labeled, for instance, under the globally harmonized scheme, which you see there. And when we validated the local lymph node assay, what we did was to show that we could differentiate chemicals in groups 3 and 4 from chemicals in groups 1 and 2. And that, in a sense, is still the underlying principle behind validation of skin sensitization alternatives at present. Groups 3 and 4 are weaker and stronger sensitizers, and of course we would like methods that also can distinguish between different potency categories. Now, having mentioned alternatives and validation, let me move on to the next slide just to show you in a few minutes what is going on at ECVAM, the European Centre for Validation of Alternative Methods. On the left-hand side of this slide, you see 3 methods mentioned. In short, they call them MUST, the HCLAT, and DPRA, the peptide reactivity assay from Procter & Gamble. And these methods were submitted a couple of years ago by the European Cosmetic Association to ECVAM, and ECVAM has been undertaking a pre-validation process, which I'll show you in just a moment. But actually what we're going to focus on more in this webinar is KeratinoSens, a HACCP cell-based assay with a reporter gene system, and I need say no more about that because I know that Kim is going to give you all the detail of that. But all 4 of those assays are actually under ECVAM review in various ways at the moment. So first, in the next slide, let's look at those 3 assays that were on the left-hand side, and I apologize for the complexity of this slide, but if you focus for a moment on the left, you will see the 3 assays, this time with peptide reactivity at the top, and then the 2 dendritic-like cell line assays, the MUST and the HCLAT from France and from Japan, on lower down. The left-hand part of the slide where all the green dots are saying completed was all about the development of the validation process within ECVAM. If you like, it was getting all the theory and the paperwork in place. Then in the middle you can see a little list of the laboratories that are participating in the validation trial, which of course involves, I should say, between and within laboratory reproducibility of these assays. It doesn't focus in any great detail on predictive accuracy because that has to some extent already been done. On the right-hand side, you will then see the different phases. The training and transfer was phase A, that's all been completed, and the proper blinded validation trial that is going on at the moment is represented by the phase B, stage 1 and stage 2. And you can see that for 2 of the assays we're quite close to completion, and especially for the peptide reactivity assay. A little bit more about that later. What happens though when we get to the end of that process? I can't give you any detail of the results because it's a blinded process. Next slide please. But what I can do is tell you just a little bit about what happens afterwards. When we get to the end, for each of the 3 assays, the validation management group, which is the group that Amanda mentioned that I chair, we make a formal report of the work and present that to ECVAM. What ECVAM will almost certainly want to do is to obtain a completely independent opinion on the work, which they will do from the ECVAM Scientific Advisory Committee. Now, I also happen to chair that, but because of the obvious conflict of interest, at that point, when the material comes forward, I stand down and the deputy chair takes over. And ESAT questions often come at the end of the validation process. Next slide. But what about KeratinoSens? Well, you're going to hear quite a bit more about that in the next 30 minutes or so. But it's just worth me saying a little bit about what's going on because Andreas Nash at Givaudan has worked very hard with this assay. He's done it, in a sense, independently of ECVAM, although he's taken regular advice from ECVAM. He's done an inter-laboratory trial, you'll hear about that. He's done intra-laboratory reliability, and those are the same steps that are occurring at the moment for the peptide reactivity and the dendritic cell assays within ECVAM. So if you like, these things are going in parallel but by slightly different routes. But KeratinoSens has accelerated, I think, past the ECVAM trial, you might say because it's been done independently, and they've been able to submit a great body of work to ECVAM, which is under active review. So what's going to happen in the next month? Well, in the next slide, I detail what I think will occur. Of course, I can't predict the future accurately. I can only predict the past with any real accuracy, but I think this is what is likely to happen. We know that, obviously, I know that ESAC has established a sensitization working group to provide expert review responses to ECVAM questions, and those questions ESAC will be finalizing next week at our meeting. Also, the ECVAM Validation Management Group for skin sensitization is meeting a bit later in October to review the peptide reactivity assay, because we think we will have all the results in, fingers crossed, by that stage. And then the ESAC Sensitization Working Group will meet right towards the end of October to respond to questions that have been put to it, with an aim, I think, ultimately of reporting back to ESAC by next spring. In the next slide, let me just summarize what I think that means. We can only speculate, but if the peptide reactivity results and the KeratinoSens results are both positively evaluated, which I think is reasonably likely, then they could provide very useful complementary analyses, essentially of sensitization reactivity. Sorry for the misword there. Now, there are some little limitations. For instance, DPRA misses metabolic aspects of sensitization. KeratinoSens, on the other hand, has the metabolic bits, but it might miss the lysine-reactive chemicals, although there aren't many. But if you combine the results from those two assays, perhaps they will cover the gaps sufficiently so that instead of having 80% or so, maybe 85% accuracy, perhaps they would together give hazard identification accuracy of around 90%. And in many people's minds, I think that would be sufficient accuracy to replace the local lymph node assay and the guinea pig assays. It would present a big challenge, of course, for risk assessment, because we might not get the potency data that we currently use from the local lymph node assay. But what I think it does mean, in summary, of course, is that it's a very timely point to be thinking about sensitization alternatives. And at the moment, the one that is actually just by a neck out in the lead, because it's the most advanced in terms of the data and where it is in the formal validation process, is KeratinoSens. So it's well worth staying tuned to this webinar now to listen to what Kim has to say. Thank you.
Thank you, David, for that thorough introduction. Next, we will hear some specific details on the KeratinoSens assay from Dr. Kimberly Norman. Dr. Norman currently works as a study director at the Institute for In Vitro Sciences in Gaithersburg, Maryland, where she supervises the conduct of the bovine corneal opacity and permeability assay, the KeratinoSens assay, and antioxidant performance assay. Since joining IIVS in 2009, Kim has been involved in exploring in vitro assays, which may be used to evaluate the skin sensitization potential of chemicals. Kim?
Thank you, Mandy. I will be discussing the interlaboratory investigations of the KeratinoSens assay. In this presentation, I will discuss the background and technical details of the KeratinoSens assay, also the design and results of the interlaboratory study, and the next steps towards advancement of the KeratinoSens assay. The KeratinoSens assay examines a cellular toxicity pathway which is induced by skin sensitizers, using a novel keratinocyte-based reporter cell line which was developed by Givaudan. The assay features a high-throughput, technically simple format, thereby making it amenable to transfer amongst laboratories. A ring trial has been completed and these results have been published. These results indicate a good predictive value and make this assay a strong candidate for validation to meet the regulatory deadline to replace animal tests for skin sensitization. The rationale for the KeratinoSens assay is that a feature all sensitizers have in common is their intrinsic electrophilicity or their potential to be transformed to electrophilic chemicals. The NRF2 electrophile sensing pathway, comprising the repressor protein KEAP, the transcription factor NRF2, and the antioxidant response elements, is able to detect skin sensitizers. In the diagram below, I will point out a few features of this pathway. In the induced state, electrophilic skin sensitizers, as depicted here, interact with the highly reactive cysteine residues of KEAP1, thereby allowing covalent modification of KEAP1, which leads to the dissociation of NRF2 from KEAP1. NRF2 then accumulates in the nucleus where it activates genes having an antioxidant response element in their promoter sequence, such as the NRF2 target gene. The KeratinoSens reporter cell line is comprised of the Hackett immortalized keratinocyte cell line, which contains a reporter construct with a copy of the ARE element of the human aldoketo reductase gene, which is stably inserted upstream of a luciferase gene. And the endpoint that we measure in the lab is the induction of luciferase activity by allergens. In the cartoon below, we see that once the chemical allergens interact with the reactive cysteine residues on KEAP1, NRF2 is able to dissociate, move into the nucleus, and activate this luciferase gene. And in the lab, we can measure this activation of luciferase with a luminometer. Next, I will go over the technical details of the KeratinoSens assay protocol. The cells are seated in 96 well plates at a concentration of 10,000 cells per well and allowed to attach overnight or approximately 24 hours. The test chemicals are then dissolved in DMSO or water and serially diluted over a wide range, which covers approximately 1 micromolar to 2,000 micromolar. Here we have a plate diagram for setup of the assay per plate. As you can see on the left, per plate, 7 test chemicals can be run, which are diluted over 12 concentrations for a full dose response. Here we see those doses ranging from approximately 1 micromolar to 2,000 micromolar. On the bottom row, 6 solvent control wells are included, along with 5 positive control wells, the positive control being cinnamic aldehyde, which is diluted over a range from 4 micromolar to 64 micromolar, and of course a blank well for compensation. Here I'm just showing one of the setups, but for each chemical we have 3 independent trials performed, and for each independent trial we have 3 replicates. So as you can see, there's a substantial amount of data which is generated for each test chemical at each concentration. In the next step of the assay protocol, the cells are incubated with test chemicals for approximately 48 hours. Following this 48-hour incubation, the cells are lysed and luciferase substrate added. And then the plates are read in the luminometer as shown below to our right. Furthermore, in a parallel plates are run, which is cell viability. At IIVS, we use both MTT and NRU to assess cell viability. And to our right, we see a picture of the KeratinoSens cells, and you can see that they maintain the characteristic keratinocyte morphology. For data analysis, we are specifically evaluating the gene induction as compared to the DMSO control wells. More specifically, we're looking for those chemicals which of the doses. Viability, depicted in pink and shown with the axis to our right here, we see that there is no effect on viability at all of the doses. This would be a characteristic response for a non-sensitizer. For the panel on our right, we have representative data for a sensitizer. For this sensitizer, we see at approximately the 5th dose, or between 10 and 100 micromolar, the chemical crosses the 1.5 threshold. That would be its EC 1.5 value at the point at which it crosses. When looking at the pink line, which represents viability, we see that perhaps 4-fold after it crosses the EC 1.5, there's a sharp decrease in the viability of the cells that incubated with this test article. Furthermore, with data analysis, we've established the positive criteria to identify a sensitizer. These criterion are that the EC 1.5 value must be below 1,000 micromolar in all 3 repetitions, or at least 2 of the 3 repetitions or independent trials. At the lowest concentration with a gene induction above 1.5, the cellular viability should be above 70%. And lastly, there should be an apparent overall dose response, and this should be similar between repetitions, and this is assessed by statistics. As you can see in the graph below for this typical sensitizer, the EC 1.5 value falls between 10 and 100 micromolar. Furthermore, at the point at which it crosses this EC 1.5 value, the viability is approximately 100%. And there is, you can't see the repetitions in this graph, but there was an apparent overall dose response, which is similar between repetitions for this chemical. Using this assay, Givaudan has made significant progress since designing the assay. They have evaluated more than 100 chemicals using the assay, along with peptide reactivity assay for complementary analysis. They have judiciously selected the chemicals which they evaluated in the assay. They selected chemicals which had congruent results for the LLNA, guinea pig, and human clinical HRIPT testing. They selected chemicals which would cover the important applicability domains, and also chemicals which would cover a broad range of potencies. The group at Givaudan has published these data and also the standard operating procedures or protocol for the assay, as noted in this publication, and these results indicate a good predictive value. Following Givaudan's success with the assay in-house, they wanted to further evaluate the robustness of the assay in an interlaboratory study, which is also known as a ring trial, or commonly called a ring trial. In the ring trial, Givaudan would further assess the transferability of the assay, the intra- and interlaboratory reproducibility, and predictivity. This ring trial would be an international effort with Givaudan, the lead lab, followed by BAS and Bayersdorf in Germany, and Procter & Gamble and IIVS in the U.S. For setup of the ring trial, 28 test substances were evaluated. These test substances were determined from chemicals that were identified in the Sensitive project, which is a large EU consortium to further refine in vitro skin sensitization methods, the ICVAM list, which is a list of LLNA performance standards, and from the publication Casati et al., which is also known as the XM list for skin sensitization chemicals. The ring trial would be set up, they're divided into 2 phases. In the first phase, 7 test substances would be evaluated, and this would be referred to as the transferability phase. And in phase 2, 21 test substances would be evaluated, which would be blind coded. We would further refine the reproducibility and predictivity of the assay with phase 2 analysis. During assessment of transferability in phase 1, all labs generated data on the 7 chemicals and then sent these data back to the lead lab for assessment. These assessment criterion are as follows. 1, the solvent control variability should be below 20%. Second, the dose response curve should be reproducible amongst the repetitions and amongst the independent experiments. And the positive control, cinnamic aldehyde, should have a significant induction within a specified concentration range. After passing these criterion, the labs would then be qualified to move into proceeding with testing of the phase 2 chemicals. Wrapping up of the phase 1 testing, we determined that in none of the labs that participated, no face-to-face training was required. Therefore, we found the SOP to be sufficiently detailed to perform the assay at a high level, and no significant technical obstacles were encountered. The only small issue that was encountered was that different participating labs used different luminometers and different luciferase reagents, being a flash or a glow substrate, which led to a little bit of difference in how they set up the experiment in terms of using reagent use. Overall, the method was found to be transferable, reproducible, and predictive when challenged with the phase 1 test substances. Moving on to phase 2. In phase 2, we would further refine the reproducibility and predictivity of the assay. 21 test substances were sent to participating labs by Givaudan. These test substances were blind coded. The data generated in phase 2 were used as final data, with no data being rejected. Phase 2 data were used to determine the robustness of the assay, and furthermore, how narrowly the acceptance criteria should be defined. The endpoints that were evaluated were the EC 1.5, the IMAX, and the cell viability, or IC50. Givaudan did a thorough assessment of the inter- and intra-lab reproducibility and an assessment of predictivity as compared to the existing LLNA data for those chemicals. Here I have an example of the published data from, this is actually from phase 1. Here we see a table which contains 6 graphs. These graphs represent the dose-response curves for dinitrochlorobenzene, which is a strong or extreme sensitizer. These 6 graphs represent the data from the lead lab in the ring study, the lead lab historical shown here, and then the 4 participating labs below. As you can see, both the lead lab and all the participating labs show similar dose-response curves, in that looking at the blue line, which is for induction of the luciferase gene, each lab shows that at a very low dose, we see it crossing the EC 1.5 as shown here on the y-axis. And this EC 1.5 value is similar for each of the labs. One difference you will note is the dynamic range of the assay, or the IMAX. For instance, here lab 1, it goes up only to about 4, and here it's off scale. And so the dynamic range was something that we found that was different amongst labs, which was related to which luminometer was used, and also whether you used a glow substrate or a flash substrate. So overall, these results appear to be very similar between labs, both for the induction as represented in blue, and for the viability as represented in pink. In this next slide, I'm also showing more of the published data. For now, we are looking at the chemical citral. Citral is more of a moderate to weak sensitizer. And once again, we see that if you're looking at the blue line, each lab shows a similar dose-response curve for this chemical. Looking at the pink line representing viability, each lab also shows a similar response for the IC50. In contrast to the previous data set looking at dinitrochlorobenzene, which crossed at a very low concentration, which was a strong sensitizer, citral crosses at approximately between 4th and 5th dose. Perhaps this indicates that this assay could be used for some materials to assess potency, and this is currently under evaluation as to whether we can assess potency based upon the EC 1.5 value. Lastly, I'll showing some published data which represents a classic non-sensitizer, or sulfanilamide. As you can see for the dose-response curves for each lab, no lab has a dose which crosses that 1.5 threshold, indicating that this material is a non-sensitizer. Furthermore, in the open squares, which represents viability, no lab shows a sharp or any decrease in cell viability really over the entire dose response of 12 doses. In this slide, this is a compilation of all of the data generated in the ring trial. As you can see, based upon color, there are congruent results for most chemicals, both within lab and between lab. So just to set up how to look at this graph, as we see in the top panel here, all of the sensitizers are listed. Those sensitizers which caused positive results are shown in red, with here, for instance, 3 of 3, meaning that in each independent trial within each lab, the chemical shown to your left was identified as a sensitizer. So we can see that most of the sensitizers were identified as sensitizer in all of the independent trials in all of the labs, showing strong inter- and intra-laboratory reproducibility. However, as you can imagine, when generating this amount of data, there will be some outliers and some incongruent results. And I'd like to just point out one of those. Our top chemical here, hexylcinamic aldehyde, which we know is from the historical data, and Givaudan is sometimes gives mixed results. And we found that even within labs, the results were not always congruent, and they weren't congruent across the various labs that participated. So moving on from the sensitizers, I would like to discuss the non-sensitizers shown here in the bottom. And for these, we would look for, all of these should be in green, meaning that in 0 of the 3 independent trials was the chemical identified as a sensitizer. So we see that for most of these chemicals, they were never identified as a sensitizer, and you see very congruent results both within lab and between labs. For these, I'd like to point out one material, the sodium lauryl sulfate, and we found that this material, although it did cross that 1.5 threshold, it tended to only cross that threshold at concentrations which were cytotoxic, which had a level of viability less than 70%. I'll give you a minute to look through some of the chemicals on this graph, as it contains a lot of information. So wrapping up the ring trial, overall we found, and the other participating labs found, the KeratinoSens assay to be transferable, reproducible, and predictive. The predictivity rate or accuracy rate was also similar amongst the labs that participated, with the range being from approximately 85.7% to 96.4% accurate as compared to the LLNA data. The EC 1.5 value had a low variability both within lab and between labs, whereas the IMAX was more variable for reasons that I had discussed earlier, such as the dynamic range of using the various substrates being a flash substrate or glow substrate. Furthermore, the IC50 was also similar within lab and between labs. The assay was found to be cost-effective and time-efficient, and these data have been published in vitro toxicology. So the next step towards validation, Givaudan is seeking validation through ECVAM, as David shared with us earlier, and the KeratinoSens assay has been accepted for review based on the data presented to date. At this point, Givaudan seeks to advance the assay through several methods. One, delivering a GLP method for broader use, generating additional data to expand the database, thereby refining the predictivity and the applicability domain that may be covered by the assay, and then publishing these additional correlative data with the correlations to the human data or LLNA or the guinea pig maximization test. In order to advance the assay, Givaudan has set up a collaboration with IIVS. Under this agreement, IIVS is able to provide research services to third parties or clients on a fee-per-service basis. The clients are then encouraged to share the non-confidential data that are generated in these studies with Givaudan for publication or for further refinement of the predictivity of the assay. The confidential data, which will be determined by the client, will not be shared with Givaudan. Furthermore, under this agreement, IIVS informs clients that the results generated through these studies at IIVS may not be published without the consent of Givaudan, and that patenting any derivative of the KeratinoSens cell line or the assay is prohibited. Furthermore, in the future, we seek to have this assay available through additional sources. At this point, I'd like to acknowledge those that were involved with generating the data that I've presented in this assay. Here I have the list for all those authors that were a part of the publication in vitro toxicology. Furthermore, I'd like to thank our collaborators at Givaudan that were not involved with the lab, Greg Adamson and Graham Ellis, in addition to Andreas, which was our primary contact at Givaudan. IIVS would like to especially thank Colgate-Palmolive Company and the Clorox Company for funding our efforts in the ring trial. Lastly, I'd like to thank the IIVS team that was involved with generating the data here of Heather Inglis, Allison Hilberer, and our management at IIVS, Roger Curran, Hans Robbie, and Aaron Hill. Thank you for your participation, and now I'll turn it back over to Mandy.
Thank you, Kim. We will now open up discussion to address some of your questions. Joining us to answer your questions is Dr. Andreas Natch. Dr. Natch works in the research department at the fragrance manufacturer Givaudan. Due to the high regulatory pressure, developing and implementing in vitro assays has become a key focus of his research in the last 5 years. So welcome, Andreas.
Welcome.
For our first question, it is, when will an in vitro test for skin sensitization be required for cosmetics for regulatory purposes? 2013 or later? This is likely for David.
These are always challenging questions, and sometimes I like to precipitate a little bit of debate, Amanda, but the strict answer, of course, still is that it's the March date in 2013 is when a replacement should be in place, because there's been nothing which so far puts a delay onto that time slot, although that is and remains possible. I think the other way of looking at it that does sometimes stimulate a little debate, of course, is that there is no regulatory requirement for a skin sensitization test for cosmetics, because the regulations that apply to cosmetics are not the same as those that apply to chemicals, pesticides, drugs, and so on.
Okay.
And I apologize if that's a confusing answer, but there is no regulation at the moment for cosmetics which says that you must do a local lymph node assay. What you might have to do is provide information on sensitization, do some risk assessments, but how you achieve that is up to individual companies and their good toxicological scientists. All that will happen in currently March 2013 is that you should no longer be using local lymph node assay or other in vivo data, and in theory you shouldn't even be using that data if it was generated for a different purpose other than the cosmetic use of a substance. But that's almost a legal conflict which remains to be.
Chemicals that are not water soluble. I'm assuming this is in the KeratinoSens assay, so this is probably for Kim or Andreas.
Yes, I may answer this question. Yes, there is a limitation. What we've seen is chemicals with a C-log P above 5, we cannot reasonably test. We may still test them at lower concentrations, but we will not be able to test them up to the high concentrations in which we test other chemicals. So this is a limitation for a few chemicals of very, very high hydrophobicity. Nevertheless, we can test many quite hydrophobic chemicals because we have solvent concentration of 1% in our assay, which is quite high for a cell-based assay, but which doesn't affect the assay.
Okay. Here's another one. What is the turnaround time for data generated in the KeratinoSens assay?
I'd like to take that question from a guest perspective. Currently, we run the assay with 3 independent trials performed by 3 technicians in this week, each technician performing 3 replicates of the assay. And so we can evaluate 7 test chemicals per week the assay.
Okay. And I have one more question here at this time. How does the KeratinoSens assay compare to other in vitro skin sensitization assays as the HCLAT in terms of accuracy and predictivity?
Yeah, I would say based on the published data, the accuracy figures are quite close. So I think there are, for both assays, the published data indicate accuracy of around 85%. We see that HCLAT detects some of the chemicals which KeratinoSens does not detect, and vice versa. So it's not completely the identical chemicals which are correctly identified, but the overall accuracy looks pretty much the same. I think what is different between HCLAT and KeratinoSens is that we normally see a signal which occurs at subtoxic concentrations, whereas for an HCLAT you normally need to go to partly cytotoxic levels to see the induction of the markers.
Okay. Another question. The 1.5 value, or 50% induction, is stated as the value for determination of sensitization, but the potency was also brought up. It's important to determine potency of sensitization as well. Okay, more Andreas.
So I can address that. Currently, the assay is used for the identification of sensitizer versus non-sensitizer. It's currently under evaluation whether we can use the EC 1.5 value to address potency. For some chemical classes, it doesn't address potency properly, but for others it does. As you can see from the graphs I had shown with the dinitrophenolbenzene, which is a stronger extreme sensitizer, that the EC value is very low, whereas for the weaker sensitizer, citral, it was much higher. So right now it's just, I guess, still under investigation whether this assay will be able to evaluate potency.
And Kim, if I can add into that, David Basket speaking, one of the other issues about evaluation of the ability of any assay, even the local lymph node assay, to measure or estimate potency is that there is only a relatively limited amount of human information on the intrinsic human potency of sensitizers. So we really do lack a gold or even a silver standard potency data set against which to judge things. I think there are some efforts on the way to try to address that, but we're certainly not there yet.
Okay. And we have another question. The LLNA is often criticized for generating false positives with surfactants. Has the KeratinoSens assay been adequately assessed for this yet?
Yes, I would say we started this especially with surfactants, false positive surfactants, and you may have seen the publication of Ball et al. In Regulatory Toxicology and Pharmacology, which just appeared a couple of months ago, where we did compare 9 exemplary surfactants. And for this set of chemicals, I would say yes, we don't see these false positives. We occasionally see gene induction, but it's always at the cytotoxic level, as Kim has explained for SDS. Now, certainly there can be other false positives which have been mentioned in the literature, and we will need to address also those classes of chemicals. But for the surfactants, I think we have shown it quite nicely.
Okay, then are the results comparing the KeratinoSens assay with the LLNA guinea pig assay published in that article that you referenced, the Toxicology Applied Pharma 2010?
Yes.
Okay. All right. Here's one that I believe is for David. Could in silico methods become a part of an integrated testing strategy for skin sensitization?
All right. It feels like a flippant answer, but the answer must be yes. And surely they are already part of integrated testing strategies for skin sensitization. My own view is people should be taking outputs, whether it's the guinea pig or the mouse or whatever, and using those outputs in combination with any other available information, which might include, of course, information from structure, activity relationships, structural alerts, and so on, and integrating the whole of that to make a judgment on substances. And I think that should continue, and I hope will be enhanced as we move forward. But I have to say, looking at a recent presentation that I saw in Brussels last week, I was certainly found myself questioning the real utility currently of the OECD toolbox, which seems to be not only in a constant state of flux, but also rarely better than tossing a coin, frequently worse than tossing a coin, at least when it came to sensitization. So one really does have to, I believe, take some of the in silico tools and use them with caution, and make sure that you not only use the in silico tool, but also the little gray cells in your brain at the same time. I think that's what makes a more powerful adjunct to the in vivo and in vitro assays.
Okay, here's one for Andreas. Why report an IC50 when 70% viability is used as a cutoff?
Yeah, that is because it's so conventional to report cytotoxicity level as IC50s. Actually, what we do calculate in the automatic calculation template, we also calculate the IC70. And so if a client wants to see the IC70, this is very easily reportable because it's always calculated.
Okay, and here's another one. How was an induction cutoff of 1.5 selected?
Yeah, I would say that was selected empirically, because we've seen when testing a number of chemicals that some of the very weak chemicals, I may mention phenamic alcohol, for example, they don't go above a two-fold induction normally, but they get a very stable induction between 1.5 and 2-fold. And so this threshold was selected also because the gene induction at 1.5-fold is normally statistically significant, and only if it's statistically significant, these data are taken as positive. And I think that EC 1.5 seems to be quite good for the yes-no answer. But what we also see is that when we look at some potency correlation, it might also be another one, like the EC3 or the EC 4.5 we have been using when we do comparisons of potency. So these other induction factors can also be calculated to give an idea of the dose response, and to use that in an integrated potency assessment.
Okay, while we have you answering questions here, Andreas, I think I have another one for you. Have metals been evaluated in the KeratinoSens assay, and what were the results?
Yeah, we have not evaluated many metals because in general we think metals are not very important for a predictive assay because nobody develops new metals. We know which metals are there and which ones are sensitizing. But we have evaluated, so for example, cobalt chloride, which is a well-known sensitizer, is clearly positive. And we have evaluated nickel chloride. Nickel is positive. It is very close to the cytotoxic level positive, but it's still positive according to the prediction model. And this has also been reported before that nickel is an inducer of the NRF2 pathway. We haven't tested non-sensitizing metals so far. I wouldn't make a statement on the predictivity for metals, but we don't consider it as a very important aspect.
Okay. Has surface area of application also been considered in this assay?
Is that meant in a way of dose per unit area? I don't completely understand what's meant with those with surface area.
The question reads that since this is also a determinant of sensitization potential, they're wondering if the surface area of application was considered in this assay.
No, we really look at concentrations in the solution when we do our quantitative data. I mean, certainly this is something where we need more statistics to see how this translates. And we are sure it will never completely translate into an in vivo potency measurement. So there is, and that's the big gap which also David correctly mentioned before, where when we go to transfer it to risk assessment, we will need read across. We will need to compare potency to potency of things where we have a clear idea of the dose per area which is sensitizing.
Andreas, I completely agree with that, and that's the way that you will translate information from an in vitro assay or an in vitro ITS across into something that we understand is a bit more tractable and where we can use the micrograms per square centimeter information which is of course critical for risk assessment.
Okay, I believe this one might be for Kim. How many chemicals can be tested in a typical run, and what are the costs?
In general, we can test up to a full plate, as I showed the plate set up earlier. So we can test 7 chemicals per week, and that's with 3 technicians running independent experiments, and each technician having 3 replicates. So we can evaluate up to 7 chemicals per week. And in terms of the cost, we do offer some economy of scale pricing with higher material, with a larger number of materials, and the cost is determined on a per project basis and also dependent upon the client needs in terms of reporting, etc. But I can say that from my research, this assay price is lower than the price of a traditional LLNA, and it's comparable or lower than the cost of an HRIPT test.
Okay. This looks like it might be going back to Andreas. What is the rationale for 48-hour incubation time? Is the 48-hour time point sufficient to predict sensitization?
Yeah. So we've looked at the kinetics when developing the assay. So 48 hours certainly is a relatively long time, but it's the same as for example, in the MUST assay. And what we see is the luciferase signal goes up very clearly after 24 hours. So for most chemicals, we could read it at 24 hours. We've seen that for some weaker allergens, it takes a longer time, and that's why we determined the 48 hours as the best time point. When we look at the RNA level, when the luciferase gene is turned on at the RNA level, we see that already occurs after 8 hours. But it takes some time for the luciferase at the protein level to accumulate. So that's why we can only measure at 24 hours, but to get a good accuracy also for the weaker ones, we selected the 48 hours endpoint.
Okay, why is the dosing done in micromolar instead of grams per liter? And the reason for the question is that a MW cannot always be derived from more complex chemicals like mixtures or polymers, which would remove this type of chemical from the applicability domain of the test method.
Yeah, no, what we do, we certainly can also test chemicals at the milligram per milliliter level, and we do that. And in this case, what we do to select the maximal dose, it is we select the maximal dose as it would be a chemical with a molecular weight of 200, which is the average molecular weight of the sensitizer, so that we have the same micrograms as a maximal dose used. But then we do all the dosing and reporting in a microgram per milliliter way. Because of course also for the LNA you can always translate the percent to molar and vice versa, as is often done in scientific literature.
And I can add to Andreas's answer in that here at IIVS we've had other individuals asking those same questions about, for perhaps these mixtures of chemicals for which a molecular weight is not a suitable measurement. And we do run those, as Andreas said, on a microgram per mil basis, just calculating back to the molecular weight. And we've begun to evaluate those materials here. And we look forward to defining the predictivity and the applicability domain to be suitable for those types of materials.
Are there limitations for chemicals that are cytotoxic?
Yeah, what we see, I mean, many chemicals, many sensitizers, especially the strong sensitizers, are highly cytotoxic, as we see, for example, in the graphs which Kim has shown for D and CB. And this is not per se a limitation. So because often these strong sensitizers are toxic at low concentrations, but even induce the gene under even much lower concentrations. But what we see, there are a few chemicals which are quite cytotoxic, and then the cytotoxicity really starts below the level when the gene could go up. So that can be for things which are very strongly membrane toxic, like a long chain acrylate, for example. And that is a limitation for very toxic chemicals, but which are not very reactive. This is an unusual group of chemicals. But in general, cytotoxicity is not a limitation.
Okay, and we have time for one last question. Have any studies been done on mixtures?
We are currently evaluating that, but we haven't anything finalized on that.
So once
and
again,
I think mixtures are also difficult in animal tests.
Yes, and as an addition to Andreas's answer, at IIVS we have run some compositions which are mixtures, and I've begun to compare the KeratinoSens data back to the other available data for those mixtures, be it the human data or animal data. And at this point, I'm seeing a nice correlation, but these data are unpublished.
Okay. I'd like to thank our panelists and all of the attendees for your participation in the webinar today. We hope that you've gained a better understanding of the status of non-animal testing methods for skin sensitization and the KeratinoSens assay in particular. We would like to remind you that questions not answered during the Q&A time will be addressed by the panelists, and the answers will be disseminated to all of the webinar participants within a few days. Please keep an eye on your email and our website, www.iivs.org, for announcements of future webinars. Thank you once again for your participation, and we hope to see you again soon at a meeting or during our next webinar.